Welcome back, Biology of Cells and Tissues. This is our lymphatic system part 2. Jumping right into the secondary lymphatic organs. The first video we talked about the primary lymphatic organs as well as the tissues, a bunch of other stuff. If you haven't watched that video, jump back to that one first before we get rolling with this one. Okay, so secondary lymphatic organs. First thing we're talking about are the lymph nodes. Okay, so the lymph nodes, if you remember right, we mentioned these briefly, that they are found along the lymph vessels. So the lymph vessels, we're going to have a lymph vessel kind of run into our lymph node. The lymph node is going to be responsible for doing a lot of that filtering and stuff that we want them to do. So then the lymph is going to continue out and continue down that vessel path. Of course, we have afferent and efferent vessels, or afferent and efferent parts coming into and out of the node. Always remember those words. Afferent is carrying it towards something. Efferent is carrying it away from something. Remember, we always talk about the two different things. We talk about stroma, we talk about parenchyma. We're just going to hit some of the important structures and look at the general anatomy of these kind of things. So there's a network of cells that support the structure and function of lymph nodes. Again, that's going to be the kind of same general definition of stroma for all of these structures. Part of the stroma, we have what is called the hilum. This is this whole area right here where we have our veins and our arteries and stuff coming in and out of the vessel. This is where arteries and nerves enter, and then veins and the lymphatics exit. Very similar to what the kidneys looks like as well. This, you can almost compare the lymph nodes to very mini kidneys. Just like the kidneys are responsible for a lot of filtration and that kind of stuff, the lymph is doing a very similar job to the kidneys, just at a much smaller level. We have a couple more structures, the capsule and the trabeculae, just like what we talked about with the thymus. Capsule on the outside, trabeculae going into it. We have a subcapsular sinus and also peritrabecular sinuses. This is different from the thymus. Remember, the thymus did not have any sinuses, but this one does. So these sinuses we can see right here. The sinuses are these little spaces. So we have right here lymph flow comes in, and we have these subcapsular sinus all the way around, and then down deep to the trabeculae we have these trabecular, the peritrabecular sinuses. These are just those main points that we talked about: the hilum, the capsule, the trabeculae, the peritrabecular sinus, and then the subcapsular sinus. Talking about the parenchyma now, remember this is the active portion of our organs. We have split into three different levels: cortex, paracortex, and then the medulla. And the medulla is made up of medullary cords and medullary sinuses, which we will zoom in and see some of that. So the cortex contains nodules of B cells. The paracortex is an accumulation of T cells. Remember, with T cells, they're produced in the bone marrow, but then they go to the thymus and they mature in the thymus, and then they can be released from the thymus into the lymphatic system, and they can be stored in parts of the lymph nodes. And always remember, the B cells, remember, they do form nodes/nodules. T cells do not form nodes/nodules. So here are our different structures. The cortex is the outermost portion. We have then the paracortex, run that inside, and then the medulla smack dab in the deeper part in the center, where we also have the medullary sinus and the medullary cord as well. So we're going to go through each of these components. So talking about the cortex first, again, this outermost layer, we have two different types of follicles. Okay, we have primary follicles and we have secondary follicles. The primary follicles, big thing to remember, these are mostly B lymphocytes. And these cells have not been exposed to an antigen. We should say the word antigen. An AKA that you might hear is agglutinogen. Agglutinogen, I think that's how it's spelled. That's just an AKA for antigen, but it is the component on the cell. So every single cell has little protein markers. So for example, like a red blood cell, we have like a red blood cell. We will have these little proteins that stick off the edge. They're like little identifiers that stick off of cells. Whether that cell, be a normal body cell, or whether it be a foreign cell. Bacteria have these markers. Pollen has these markers. Anything, any kind of substance is going to have these little protein markers like this. In blood, eventually we'll talk about this in the future, but blood, these little protein markers that we have out on the edges of the cells, they determine our blood type. And this isn't really necessarily important for this specific lecture, but it will come around in the future eventually. So these little proteins, we also call them antigens. Antigens meaning that they are going to be what stimulate the immune system, immune response, if they are not recognized as our own antigens. Okay, so we're looking for foreign substances and that are not supposed to be there, that we will want to attack and that we will want to destroy. So it's not been exposed to an antigen like that, so it's not going to be active. Then we have secondary follicles. So this is when the primary follicle encounters a foreign antigen, specifically a foreign antigen. It then transforms into the secondary follicle. It contains lightly stained central region. The primary is densely stained. And these are the B cells responding to an antigen by proliferating and differentiating into the plasma cells, which are then going to produce the antibodies. So here we have a little bit of picture. You can see the secondary lymphoid follicle versus the primary lymphoid follicle right there. And here is our pictures of it. So the primary lymphatic follicle, again, she calls these nodule or nodes. I prefer the term node. Follicle is the more correct term, but any form like this is going to be fine when it comes to test purposes. Follicle, nodule, node, it doesn't really matter. So here we have all clumped up just like this, all in through here, mostly these B cells. We're going to see uniformly dense and stained quite dark on this picture. Is that our primary follicle? Our secondary follicle, we have a lightly stained central region because this is now responding to the antigen. It's proliferating and differentiating or changing into the plasma cells that it needs. Here's just a couple more pictures of the lymph nodes. We can see we have cortex. Here's some follicles. We have a capsule. We have the hilum. Medulla here in the center. We have the paracortex. I would 100% use this picture specifically when prepping and studying for the lab portion, as this is the kind of stuff that she'll like to point to. I would make sure you recognize these guys. Okay, what are these structures in here? The whole structure is a lymphatic follicle or a lymphatic nodule, a primary lymphatic nodule, pardon. And then what's it made up of? It's made up of a bunch of B cells. Here's another picture. We have cortex, we have paracortex, and then we have medulla with some of the other portions in there. Okay, so that's kind of the makeup of the lymph nodes. The next important thing that we want to discuss is actually the circulation through the lymph node, both from the lymph circulation going through it, as well as the blood circulation that goes through it. So, starting off, we have our afferent lymphatics, our afferent lymphatic vessels. Then we move, so we see they're coming in, these afferent ones. They go into the subcapsular sinuses. They continues to flow down around the trabeculae, so the trabecular sinuses, the peritrabecular sinuses, until finally it goes into the medullary sinuses towards the center. And then as it's filtered all throughout there, and then it goes out through the efferent lymphatic vessel. You'll want to know just that step by step what the flow looks like. Next, we talk about blood flow. So with blood circulation, we have arteries that enter at the hilum. Then they branch through the stroma. Remember, which stroma we're talking about, like the skeleton, all the supporting cells, all the area around the active cells. Then it goes down into little capillaries inside of the cortex. And then it gathers again into venules with they're called post-capillary venules in the paracortex. And then the veins exit at the hilum. And you can see I have the arrows coming in through here, coming all the way down. Let me actually finish these arrows. I'm going to use a different color, though. I'm going to use just this blue kind of color. Actually, never mind. I already have yellow. I just didn't notice it. So you can see here the black coming in through the arteries, and it splits off into capillaries inside of the cortex, and then the veins converge to venules, and it continues on its way out. Again, you just really want to know kind of that step by step. And it's within these chunks with the let me see. This is just another picture of the lymph nodes, a reminder that these are filtering all this stuff throughout. This is just a picture of that post-capillary venule of the lymph node within the paracortex. They call them HEVs, or high endothelial venules, found in the paracortex. I wouldn't worry too much about that fact. This is probably going to be one of those one-off test questions that you may or may not see. Something along the lines of, where are the HEVs found? The answer being the paracortex of lymph nodes. That's a question, if you want to get an A in the course, you should know that. Or if you want to get an A on the test, you should know that specific little fact. But not knowing it, you're still more than likely going to get a high B if you focus on like the rest of the stuff. Clinical application. So this kind of goes with lymph nodes, and if you know anybody who has cancer, especially breast cancer is a big one. Where usually wherever they find that cancer, when they go in and they surgically remove it, a lot of times they remove the lymph nodes in the surrounding area as well. And that's because it's through the lymph nodes that you get a lot of metastasis or spread of cancer cells. So like metastatic cancer cells that detach from a primary tumor can enter the lymphatics and are carried to nearby lymph nodes, especially what's known as the sentinel lymph node. Sentinel lymph node is just saying that it's the first lymph node that's downstream from the tumor. Okay, so like if I have a tumor, like if you take somebody who has breast cancer, they have a tumor in their mammary tissue, most often the first chunk of the first lymph nodes are going to be within the axillary lymph nodes, because they're right there. So a lot of times you'll see if somebody has breast cancer and they go in and they surgically remove it, they'll also remove those axillary lymph nodes as well, just to be safe. to make sure that there's no spread of any cancer cells, just like in case that they miss anything. So this is also determined the metastatic, how spread the cancer is determined by, it determines the staging, as well as like how spread it is within the lymph nodes determines the staging system as well. Lymph nodes, again, lymph vessels in the lymph system are super important, and you can have, I think it's mentioned in, I think it's in either physiology or general pathology, or potentially both. I know it's a little bit future courses as well. They start to talk a little bit more about the lymph system, and they mention, if you have lymph nodes removed, obviously there's a difficulty then moving the lymph through, because you also have to remove those vessels. So people who have cancer, have issues like this, oftentimes like if they fly or do things like that, they have to wear like compression sleeves, or like put their arm up or their limb up in order to help that lymph flow back to the center of the body. There's also like some parasite. There's a parasite. It's microbiology next trimester. In microbiology, you'll talk about this. There's a specific parasite that likes to go and enter the lymph system, and it causes a blockage in the lymph system, which causes massive swelling. So you'll see those kind of things in the future. Okay, the next organ is the spleen. This is not to be confused with the pancreas. Make sure you remember pancreas and the spleen are two separate organs. They look very similar, but they are very different from each other, okay? Okay, so this is our largest lymphoid organ. It is responsible for filtering the blood. It destroys our old red blood cells. It produces activated lymphocytes in response to blood-borne antigens. And it's made up of two kinds of pulp, which we call red pulp, and we call white pulp. Red pulp because blood, white pulp because lymphocytes. Again, we have the stroma, we have capsule, we have trabeculae, and it's made up of reticular connective tissue. The parenchyma we're going to get into, okay? We have white pulp, we have red pulp. The white pulp, I'm going to hit with a little bit of terminology, and then we'll talk and we'll see each of these, okay? So the lymphoid tissue surrounding central arteries is known as the white pulp. Central arteries are a specific type of artery that we will see. There are periarteriolar lymphatic sheaths, or PALs, composed of T cells. And actually, technically, this should be indented like this. So that lymphoid tissue that surrounds it, there is a periarticular lymphatic sheath, a periarteriolar lymphatic sheath. So it's essentially this coating, made up of T cells that surround these arteries in the parenchyma of the spleen. And then you also have these splenic nodules or nodes, and that are full of B cells. I'm going to find the right. There's a nice picture. Well, when I find the picture, we'll go into it. Then we have the red pulp. The red pulp is the tissue that filters the blood, removes waste, and destroys the bacteria, viruses, other stuff that needs to be taken care of. There are these structures called splenic cords, also known as cords of Billroth. They're made up of reticular cells and fibers. They contain macrophages, lymphocytes, as well as other blood cells. And these heavily filter the blood. We have another structure called sinusoids. I'm actually going to delete that picture because I describe this in the next one. These are elongated endothelial cells. They transversely are arranged. They're transversely arranged reticular fibers. They have a bunch of macrophages and an incomplete basal lamina. These are kind of its own thing. So you have these sinusoids, these special little kind of like ballooned up parts. You can almost think of it like a ballooned up part of a vessel that has lots of cracks and openings to it. So they are sinusoidal, like that picture. Let me actually bring that picture back. Like that sinusoid like we talked about with the capillaries. Very similar to that. But they're ballooned like this in order to create those big gaps. So they're wide, tortuous blood vessels, tortuous meaning kind of like twisting, in the spleen that filter blood and remove damaged or old red blood cells. Okay, so that's what these guys look like. And we're going to walk through kind of the more important part of the spleen, talking about, I should say the more confusing part, which is talking about the actual blood flow itself. So let's talk about blood going into the spleen. From the beginning, we have a splenic artery, which goes into the spleen and then branches off into a bunch of smaller arteries called trabecular arteries. When these trabecular arteries reach the white pulp and become covered with pals, that is those periarteriolar lymphatic sheaths made up of T cells, they are then called central arteries or arterioles. It's used interchangeably. So think splenic artery, branches become smaller and turn into trabecular arteries. When the trabecular arteries go into the white pulp, then they are called central arteries. The central arteries branch into even smaller arteries called peninsular arteries or arterioles, again interchangeable. And then these continue to flow and get even smaller. They branch again, continue to get smaller, and are the arterial capillaries, which we can see here. Arterial, these guys are our arterial capillaries right in here. Small capillaries. So our order, we go from the splenic artery to the trabecular to because they become central, peninsular, and then capillaries. When we talk about this blood supply, we say that up to this point, getting to the capillaries, we have what is called closed circulation. This will more than likely be a test question. So closed circulation just means that the blood is always within or inside of some type of vessel, whether that be arteries, capillaries, or any other kind of vessel, to make sure that the flow is regulated tightly. Okay? So up to this point, it's closed circulation. After this, so after the blood leaves the capillaries, we are going to call it open circulation. So this is the process where blood leaves the vessels and flows freely into the splenic tissue, being that red pulp, specifically into these structures known as splenic cords. I'm going to kind of go over this a little bit more in depth in a second. Let me jump to the picture. Where's the picture that I want? Here we go. So here we have trabecular artery, so splenic artery coming in, splits trabecular artery. When it gets to that sheath, that pals, it's now the central artery and goes through. Here's our white pulp made up of all of those immune type cells. And then it splits peninsular arteries. And it gets smaller and it runs into capillaries. And see how these portions, we saw it in this picture too, it kind of runs into nothing. Like there's no continuous vessel that it attaches to. This is where the filtration happens. these structures, the structures all throughout around here, known as the splenic cords, I kind of picture it like if you think about the movie Finding Nemo, when they get swallowed by the whale and you get to see like the nice brushes of the whale's mouth, like a super filters and it filters through really, really finely. That's what I imagine kind of are the splenic cords. So basically, this is pushed through a meshwork of splenic cords. So the artery just opens completely and empties into the red pulp. And these splenic cords it's like this net that red blood cells make their way through. Healthy red blood cells can get through because they're still flexible and they're still good to go. Older red blood cells lack a little bit more of, they lack some of that, what's it called? That protein, name of the protein slipping my mind right now. Hopefully some of you guys remember the name of it. The name of that protein that helps keep them super, super flexible, because they're older at this point, so they get caught in the net. Okay? And I tried really hard to find a good picture of this, but I really just couldn't. Hopefully that makes a little bit more sense about what's going on. These vessels, or the red blood cells that are healthy that make it through, they can flow through and make it through that meshwork of those splenic cords and make it to the sinusoid and then slip past the walls and enter back into the bloodstream. And that's how it's filtered out. I think I wrote it. Yeah, I thought I wrote it in here somewhere, but okay. And then once it makes it through the splenic cords and it's in the sinusoid is further lined with macrophages, which further filters the blood and eats all of the stuff, eats any of the extra gunk that's in there. Another little tidbit. Again, this is kind of just like the mastery portion. If you want to bump your grade up a little bit higher. The cells of the sinusoid, this special type of endothelial cells, they're called stave cells. So the sinusoids are made up of stave cells. Okay, so that's the arterial side of everything. It goes through, it gets released out, the blood gets released into the red pulp, it has open circulation, and then it's collected by those sinusoids, and then it continues along its path. So it's going to go from the sinusoids, just like, thinking capillaries go back to venules, to veins, and continuing. We have then the opposite side where from the sinusoids, it collects in red pulp veins, which then continue and they converge together to become trabecular veins, and those continue to converge and become the splenic vein, and then it leaves the spleen. Here is like the overall summary of everything. This is the arterial side, kind of in the sinusoids, and then the venous side of things, all kind of broken down. And then just a couple of pictures. It helps to kind of trace this path. Take a minute, draw on this specific picture, and trace the path of the blood, how it goes through, kind of what's happening to it. Specifically, there's closed circulation here, open circulation happening here. Additionally, there's a third spot. There's red pulp, there's white pulp, and then there's a space called the marginal zone. This is a specialized area between the red and the white pulp. It's a critical role in the immune system. It is a transition zone where our blood-borne pathogens and antigens are filtered and then presented to our immune cells. The B cells, the T cells, and the macrophages. The lymphocytes these are mostly B cells. Then they can produce antibodies. Macrophages act as antigen presenting cells. And then the lymphocytes can exit the blood and enter the white pulp here as well. here's all of these components of the pictures. Got the red pulp, got the white pulp, here's the capsule. Here's one of our peri-arterial lymphatic sheaths. You can see right there. The central artery, you can kind of see it in this one too. The central artery is running this way through the middle right here. It's kind of zoomed in. Right here, so you can see a little better. Here's the central artery making its path all the way along here, surrounded by the pals, all these T lymphocytes all in through here. Here's just a close-up picture. That's kind of what the splenic sinusoid looks like. This is just talking a little bit more about those sinusoids. Okay. Finally, we have our last type of organ, which we call MALT, mucosa-associated lymphoid tissue. This is lymphoid tissue in mucous membranes throughout the body. Protects from pathogens trying to enter the body. Specifically, these are located in the tonsils, in these structures called Peyer's patches located in the intestines, as well as the appendix. We don't talk about the appendix really in this lecture. Just understand it is another organ that that's involved that has this type of tissue. So where we see MALT, one of the big places is in the tonsils. These are irregular masses of lymphoid tissue in the mucosa of the posterior oral cavity and nasopharynx, where their cells encounter antigens entering the mouth and the nose. There are 3 types of tonsils: palatine, pharyngeal, and lingual. Our palatine tonsils are paired. We want to make sure we focus on this. And they are stratified, squamous, non-keratinized epithelial cells. Bam, right there. Our pharyngeal tonsil is single, not paired. It's on the superior portion of the pharynx. The epithelium is pseudostratified columnar epithelium. These are the 2 big things that this professor had focused on, so we just want to keep those things in mind. Additionally, these tonsils, if you had heard of adenoidectomy, removal of your tonsils and adenoids. An adenoid is just the tonsils that have been chronically stimulated by infection. So especially kids who have lots and lots of ear infections, sinus infections, allergies, other sources of inflammation, anything like that, strep throat is a big one. They can get hypertrophy of the tonsils, and the hypertrophy of tonsils, then once they're hypertrophied, they then call them adenoids. And that can be an issue because it can start to cover some of the throat. It can make it difficult to breathe. It can harbor disease as well because they're enlarged. So then you can get sick more often, which is why in many cases they opted to remove them. Then we have the lingual tonsils. These are located at the base of the tongue, and there's just a whole bunch of these located there, and it is stratified, squamous, non-keratinized. Here we can see some of the tonsils. We have lymphoid nodules. There's some of the epithelium, the connective tissue, germinal centers, crypts, all that kind of stuff. Inflammation of the tonsils, also known as tonsillitis, is more common in children than adults. And then that chronic inflammation, they known as adenoids, which we discussed. And then here are what are called our Peyer's patches in the ileum, in that small intestine. These are large clusters of lymphoid follicles located there. These are very often asked about as lab questions, specifically like asking either where they're located or just to identify them. So Peyer's patches, these are groups of lymphoid nodules within the gut, within the small intestine. And that covers our lymphoid tissue. We went a little bit over in this video. It's a lot of information. Again, if you have any questions, comments, concerns, anything else going on, please drop a comment and I will get back to it as quick as I can. And have a wonderful day and good luck studying.